Yes, good evening, everybody. Good evening. Good evening. I hope you all can hear me properly. Yes, everybody. There might be some sounds from the nearby temple, but I hope it's fine with my voice. It's clear. The screen is absolutely clear. I hope my voice is clear. I hope very important chapter of your class 12th biology, biotechnology, principles and processes. And today's class we are discussing about the tools of biotechnology, lecture number 1. So take down the important points while I'm explaining in the class, and you will get the PDF of the session soon after this particular live is over. Okay, sometimes you may get the PDF in the description box of this particular lecture, or I'll provide the PDF in the group. So listen very carefully. I'll be telling a very small topic of your NCERT we are discussing today. That is the tools of biotechnology. So once again, welcoming you all to the supremely amazing, super cool class of Biopoint NEET on the chapter Biotechnology Principles and Processes. So without wasting our precious time, moving on with the lectures. Those who are watching the session, don't forget to hit the like button, do share it with your friends and subscribe our wonderful channel of Biopoint, stream the world of bio. If you have any doubts and you fail to ask to us in the live session, please do comment it in the comment section of this live so that I could explain to you in very detail in the comment section. This is an amazing Telegram group at Biopoint New where we conduct regular discussion, polls, quizzes, updates on our classes and daily OMR examination. I hope my audio is clear for each one of you. This is a crash course for NEET 2021. Do join our Telegram group at Biopoint New to know more about the crash course for NEET 2021. Yes, everybody, this is our one-shot session which we will be soon be restarting. Okay, very soon we will be restarting this one-shot session series for all the chapters of your class 11th as well as 12th, a quick revision for your NEET 2021. Give me hearts on the screen and let's move on to the chapter of biotechnology soon. If you are ready to start, if you want me to start the chapter, give me hearts or thumbs up on the chat box. Hit the chat box with a thumbs up. Yes, everybody. Yes. Can we start off with the lecture? Yes. I hope everything is clear for you. You can hear me properly. I don't know whoever is there on the chat box. Anyway, I'm starting with a lecture, one of the very important chapters. Yes, thank you so much, Calvin. So let's start with this topic, biotechnology. And first, we are going to discuss about the introduction of biotechnology. Okay, so what is actually biotechnology? Biotechnology is a technique of using the live organism or their enzymes for products and processes that are absolutely useful to human beings. Okay, so biotechnology is a technique of using live organism or their enzymes for products and processes useful to human beings. Actually, you may notice that there are certain applications of biotechnology. For example, one of the application that you all know, one of the application you all guys are familiar with is the DNA fingerprinting. DNA fingerprinting is one of the application of biotechnology. Then the method, that is the ELISA method. What do you mean by ELISA method, which is used to detect AIDS, HIV, virus and all? Okay, so ELISA method, then different tests carried out for diagnostic purposes, DNA fingerprinting, all these are different applications of biotechnology which we will be discussing in detail in the next chapter, that is the application of biotechnology. So here they have specifically mentioned you what is biotechnology. So listen very carefully. Biotechnology means it is a technique of using of different organism or enzymes from different organism for what? For producing certain things or producing certain phenomenons which are useful to human beings. Okay, for example, in your lower classes, or you might have read somewhere about the term bioweapons. Tell me yes or no on the chat box. Bioweapons. You might have learned about bioweapons, right? Bioweapons means they are certain devices, okay, they are certain things in which they are filled with a particular type of microbe, and one country uses the bioweapons attack on the other state or country or nearby continent, right? So one area will make a bioweapon attack onto the other area to destroy the entire things happening over there. Okay, yes, Bacillus thuringiensis can be called as a bioweapon, but more important bioweapons we can tell. Okay, more important, for example, in the earlier times, that is during the initial times of this COVID-19, a particular news is spread, a rumor has spread that this was a bioweapon attack by China on India. Okay, there was a particular news like that during the initial days when this virus came to India, that this was a type of a bioweapon attack by China on India. COVID-19. Okay, COVID-19 can also be considered as a bioweapon, exactly like this one Ncert, that is all the normal activities of a particular area would be getting affected by with the help of a particular small unicellular prokaryotic microorganism. Clear? So bioweapons cannot be the application of biotechnology. Bioweapons cannot be considered as an application of biotechnology. Why? Because they are not useful to human beings. It is specifically mentioned that biotechnology includes those things which comes as useful to human organism or human beings. I hope the slide number 1 is very clear for everybody. Biotechnology is a technique of using live organism or their enzymes for products and processes useful to human beings. Nobody is there on the chat box. Very good, guys. Very good. Keep it up. 0 viewers. Keep it up. Okay. Okay, so these is okay, Calvin. It's actually shown over here as 0. Okay, only Calvin is there. So listen carefully. If you have any doubt, you can ask me right now. Okay. So biotechnology is a technique of using living organism or their enzymes for products and processes useful to human beings. So let's move on to the next slide. I hope it's clear. So biotechnology means it includes only the things that are useful to human beings. Bioweapons cannot come under as an application of biotechnology. Yes. Another important thing, that is the definition of biotechnology. The definition of biotechnology, which is a frequently repeated MCQ question for your NEET examination. Okay. So the correct definition for biotechnology was given by the European Federation of Biotechnology, EFB. Okay, this is a very important MCQ. European Federation of Biotechnology. It defines biotechnology as the integration of natural science and organisms, cells, and parts thereof of molecular analogs for products and services. Definitely, exactly as how we defined biotechnology in the slide number 1. They are the technique of using a live organism or their enzymes for products and processes that are useful to human beings. So European Federation of Biotechnology have defined biotechnology as the integration of natural science, that is a combination of natural science as well as organisms, cells, parts of thereof, with all the important things necessary for the preparation of a particular product or a human welfare services. Clear? So this is a definition provided by the European Federation of Biotechnology. Well defines biotechnology. Okay. Next, we are going to discuss about slide number 3. We are discussing right now, and here we are discussing about the different applications of biotechnology, and about each of these things we would discuss in detail in the chapter number 2, that is applications of biotechnology, soon after we finish this chapter. So what are the main applications of biotechnology or biotechnology deals with? That is, this is actually the different applications of biotechnology. So what is actually applications of biotechnology? Number 1, they are used for making curd, bread, wine, etc. That is microbe mediated processes. Microbe mediated process, that is, they can be used for making curd, bread, wine. Absolutely, Calvin. The uses of biotechnology we are discussing. That is, you have to have a clear idea about where all the biotechnology finds its application, or where all we can use the biotechnological methods. Number 1, they can be used for making different products like wine, beer, then cheese, yogurt, pickles, curd, bread, wine. All these things you can use the application of biotechnology. Second one, in vitro fertilization, that is mainly in the test tube baby program. Very important. Okay, biotechnology finds its application in the test tube baby program. Okay, it is a very important MCQ question. It finds its application in vitro fertilization, that is a test tube baby program. Okay, so that is the second application of biotechnology. This is a very important statement based question that can come for your exam. Okay, in vitro fertilization finds its application in the test tube baby program. Okay, the third one is the applications of biotechnology. That is, the third one is the synthesis and using of a gene. Synthesis and using of a gene is the third application of biotechnology. That is, they can be used over here. That is, applications of biotechnology. Biotechnology finds its application in the synthesis of a particular kinds of a gene. Suppose this is one gene that we can find its application, and we have to synthesize another gene which is complementary to this. Biotechnology can find its application. Clear? Next, fourth application of biotechnology is the preparation of DNA vaccine. For example, in this COVID situation, we are finding vaccine using the application of biotechnology. Okay, so biotechnology finds its application in the preparation of DNA vaccine or any kinds of vaccine. The last application of biotechnology is it is used in the correcting of a defective gene. If there is any defection, that is any disorder in any kinds of gene, biotechnology, we can use the application of biotechnology to correct the gene. Okay, we can correct the defective gene. So the defect in the gene can be corrected with the help of biotechnology. Okay, next these are the 5 main application where we are using biotechnology. Clear? First one is a microbe mediated process, making curd, bread, wine, all these things. Second one is the in vitro fertilization. Third one, synthesis and using a gene. Fourth one, preparation of DNA vaccine. Fifth one, correcting a defective gene. So these are the main area where you can find the applications of biotechnology. Yes, next we are going to discuss about the genetic engineering. What is actually genetic engineering? Genetic engineering is a technique in which the genetic material, that is the DNA and RNA, is chemically altered. That is, the chemistry of the genetic materials DNA and RNA are chemically altered and is introduced into a host organism. That is, you can correct out a particular gene, or you can alter, if there is no need of correction also, you can alter the particular gene with the help of biotechnological methods. Is that clear? Right? So this is the first one. For example, you might have learned out a disease SCID, SCID, which is caused due to defect in the gene causing ADA, adenosine deaminase enzyme. So this is also treated with the help of genetic engineering method. Okay, so SCID is a particular disease or a defect in the gene producing the enzyme adenosine deaminase. I hope you know the spelling of the particular term, adenosine deaminase. Adenosine deaminase. Okay, adenosine deaminase, where you can find an application over here. That is, you have to discuss in detail about the concept of adenosine deaminase enzyme and about the SCID defect and all these things in the upcoming chapter of biotechnology and its application. So simply I can tell you what is genetic engineering. What is genetic engineering? It is a technique in which the genetic material is chemically altered, that is a particular gene or a DNA or an RNA, whichever present in the according to the organism, can be altered. In order to alter the chemistry or the chemical composition of that particular genetic material, they have to be introduced into a host cell so as to change the physical appearance of that particular DNA or RNA or sequence. Okay, you have to change the phenotype of that particular sequence, and for that you have to change out their chemistry. So chemical nature of the DNA sequence have to be changed. Yes, phenotype means the external feature. Phenotype definitely refers to the observable character. Okay, for example, we have to convert a dwarf person into a tall person. Look at the example. We have to convert a short organism into a long organism or a grown up organism. For this particular thing, you have to require a particular gene of interest. Okay, you have to require a gene of interest or a desired gene have to be present over here. What is the desired gene? What is the desired gene present or needed over here to convert the short particular organism into a tall one? You need the desirable gene as growth hormone. Okay, so here in this particular situation, growth hormone can act as a desirable gene because our desire or our ability or our aim is to convert a short organism into a grown up organism, and for that we have to divide or we have to insert a particular type of, we have to alter the chemistry of that particular gene of the short organism in order to convert it into a large organism, and for that we need a gene of interest or a particular thing, that is desired gene, which you call it as the growth hormone. Is it clear? That is all about genetic engineering. Okay, so particular DNA sequence is being converted by introducing it into some other organism, or that is referred to as the host organism. And for what it is mainly done, in order to convert or to change the phenotype. What is phenotype? The physical appearance or the external characters that we observe. Okay, by looking at a one particular person, we can tell that person is white in color, this person is medium white and black mixed in color, that is tall, that is dwarf. So all these are the different physical appearance. All these things can be noticed directly from the phenotype of that organism. Clear? So these are the important thing of genetic engineering. I'll show you, I'll explain it to you with the help of an example. Let me take out a blank slide. Yes. Listen very carefully. Listen very carefully over here. For example, you have given that there is a particular genetic material. Okay, a particular genetic material is given. So suppose this is a particular gene of an organism. Okay, clear? And into this particular gene, this is a particular gene for a growth hormone producing chromosome. Okay, one second, let me highlight it. Okay, yes, absolutely, it's clear. I hope right now it's clear. The diagram is clear for you guys, right? Yes. So this is a particular growth hormone. This is a human gene for GH. This is the human gene for growth hormone. We have to convert, or we have to introduce this gene, or we have to make out a particular food product containing the human gene for growth hormone. Okay, suppose a person A and B is there. A person A is very highly growth hormone is produced, but for person B it is a very short person or a dwarf person, and we needed to increase their length. For that, you have to introduce a growth hormone. There is a necessity of growth hormone in that person B. So what we can do, we cannot just take out some human gene from one person and can inject directly into the other person. One thing that we can do, we can make out a food product which can easily do them a good thing, right? We can make out a milk or a food product, especially milk or whatever food product. We can increase the amount, or we can alter that particular milk product, the quantities and the quality of the milk, by introducing a human gene also into that. But now you will be thinking that, is it possible to introduce or to mix a food product with a gene? Yes, that is what we studied in the beginning, application of biotechnology, food mediated, microbe mediated process. So I'm going to introduce this particular human gene. I'm taking out this particular gene. Okay, this particular growth into a one particular cell, into a host cell. Okay, this is my host cell in which I'm going to introduce. Into this host cell in which I'm going to introduce my. Okay, this is my host cell. And mainly you can tell the host cell can be a stem cell that is capable of division. So I'm introducing this particular gene into this particular host cell. Okay, this is the gene over here. Okay, is it clear? Next, what I'm going to do is that this particular altered gene is being introduced into a cow. I'm going to introduce this gene into a cow, and you know that cow produces milk, right? A cow produces a milk, and you know very clearly what can be the constituent. Suppose in this vessel we are taking the milk of the cow. The blue color indicates the cow milk. I'll tell you what is the final product that we obtain after this. Okay, green was our gene, right? Green was our gene. Okay, so in this particular final product, that is a milk, you will observe certain traces of the human growth hormone also. Okay, you will find milk plus the growth hormone of humans will be the final product that we are obtaining as a result of this. Is it clear for everybody? This is actually what you call it as the genetic engineering. This is actually what you call as the genetic engineering. Is it clear? Yes. So we have to desire for a particular gene. Guys, I'm not telling it is a gene for growth hormone only can be done in this way. Either, for example, if we have to treat a person with a diabetes, then this can be, we can take this as a human gene for insulin. We can take as this is the human gene for insulin. They can be introduced into a host cell or they can be introduced into the cow, and this particular here you will obtain milk plus human insulin. The gene for human insulin will be there. So a person who is having diabetes can buy and drink the quality milk in which you will have the combination of milk plus insulin. But rarely only you will find all these items in the markets. Okay, these are all under processing, but all these things are possible in this 21st century with the help of the process which is termed as genetic engineering. Clear? Whatever things we need, we can make out with this particular method. So this is how you alter the gene of a particular thing. Next, we are moving on. What are the main steps involved in the process of biotechnology? That is, I told you how you can make out a genetically modified organism or how to make up the genetically modified species. So for that, you have different things or different steps. So let's discuss one by one steps we can discuss right now. Step number 1, identification of DNA with the desirable gene. I already told you what is desirable gene. That is a gene of interest. Look, suppose we have to make out insulin. This is the human gene for insulin. Suppose, okay, this is a human gene of insulin. Look, we have to introduce this particular segment of this chromosome that produces, or this gene that produces insulin into a host organism. Okay, this is actually the gene that produces insulin. Clear? So this has to be introduced into a host organism. Second step is that, introduction of the identified DNA into the host organism. Which is the organism you can use as a host over here? That is E. coli bacteria, mainly the most common host used in the process of biotechnology is the E. coli bacteria. Clear? For example, this is a gene for human insulin. It has to be identified. So human gene with a desirable gene have to be identified first. It has to be introduced into the host cell, which is the E. coli bacteria. Can you please tell me on the chat box, why is the reason? Just have a guess. What is the reason why we are using E. coli instead of the other species of monirans? Just give a guess or a try why we are using E. coli bacteria as the common host in the process of biotechnology instead of other species of monirans. Yes, why only E. coli are we using? No idea. The thing is why we are using E. coli is because E. coli divides very fastly. E. coli divides very fastly. That is Yes, absolutely. It has the ability to multiply. It has the ability to multiply. E. coli divides in every 16 minutes. Every 16 minutes, new progenies or new generation further progenies of E. coli bacteria is seen. So, look, this particular species, that is a host organism, we have to introduce the particular desirable gene into a host organism, right? So, we have to introduce this particular 16 minutes, 16 minutes, 6 every 16 minutes. Okay. So guys, we have to introduce the identified gene into the plasmid. We have to introduce the identified gene into the plasmid of the identified organism or the host cell. Please check everybody. This is the desirable gene for insulin. Okay, we have to introduce that into the plasmid of the host cell. I hope you have been taught in the chapter cell and cells, cell the unit of life, about what is plasmid. Plasmid is the extra chromosomal material of prokaryote. Okay, we will discuss a separate class on plasmid in the upcoming class because we have to learn what is plasmid in detail. Extra chromosomal DNA material present, genetic material in prokaryotes. We have to introduce this particular gene into the plasmid. Look guys, this is the plasmid. So into the plasmid of the E. coli bacteria, we have introduced the gene that controls the production of insulin also. Look over here. You would have see that this is the plasmid, right? This is the plasmid. And in the plasmid of the E. coli bacteria, which mainly act as a host, we are introducing the gene that controls the production of insulin. Okay. So this particular new gene that is produced, you will call it as the recombinant DNA. What you will call it as? You will call it as the recombinant DNA. Recombinant DNA. Clear? Yes. Or simply you can call them as rDNA. rDNA. You can simply call it as. Right? So they have to be. We have introduced the particular gene controlling the human insulin production into the plasmid of E. coli bacteria. E. coli is the normal host we are using in the method of biotechnology. Okay. Next, the third step we have to move forward with the biotechnology is the maintenance of introduced DNA in the host cell. And we have to transfer the DNA into the progeny cell. Okay, we have to maintain the particular DNA inside the host cell. We have already transferred the DNA into the host cell. Different methods are there. For example, you can inject a particular DNA segment into the plasmid of an organism or into the host. Second, you can use certain catalyst, tungsten, palladium, platinum, and all these things to fire like a bullet this particular thing into the host organism. Or in plants, there is other methods also. Biolistic, gene gun method, all these different methods are there for introducing the DNA. So there are different methods to introduce a DNA into a host cell, but the important thing is that we have to maintain how that particular organism is maintained inside the organism under a sterile condition. Is it clear? Yes. So what are the three main steps involved in the process of biotechnology? First one, we have to identify the DNA with the desirable gene. Second one, introduction of the identified DNA into the host cell. Step number 3, maintenance of the introduced DNA in the host and transfer into the DNA progeny. Clear? Let's move on with the next slide. Yes. Let's move on to the most important thing, recombinant DNA technology. recombinant DNA technology, and we are going to discuss about who all are the persons that play an important role in the recombinant DNA technology. So let's discuss first of all what is recombinant DNA technology. We have to discuss the process of joining and inserting a foreign piece of DNA into a host organism to produce a new genetic combination is called recombinant DNA technology. That is, this is a plasmid of an organism. This is the plasmid of an organism, and you're introducing a particular gene of your interest into a particular plasmid to convert it into a recombinant DNA or a new DNA having a different or a new phenotype. Okay, so that is the important function of recombinant technology, DNA recombinant technology. So something which I missed in the slide, I'll tell you, the father of genetic engineering. You have to study the name of the father of genetic engineering. That is Paul Berg is considered as the father of genetic engineering. I'm not talking about DNA or rDNA technology. I'm telling about recombinant DNA technology. Not, I'm talking about genetic engineering right now. Back, because I missed a particular important point. Paul Berg is considered as the father of genetic engineering, and he was awarded the Nobel Prize for genetic engineering in the year 1980. You don't have to study the year, but you have to study the name of the scientist. That is Paul Berg. So Paul Berg is often called as the father of genetic engineering. Clear? And now we are discussing about the recombinant DNA technology, and now we have to discuss about the first rDNA. The first rDNA, that is recombinant DNA, was produced by Stanley Cohen and Herbert Boyer in the year 1972. Very important MCQ, very important 100% guaranteed MCQ question. The name of the scientist Stanley Cohen, Herbert Boyer, and the year also very important over here, 1972. Okay, very important. If it is not there in your NCRT, take it down. I'll provide the PDF of the session in the Telegram group. Clear? Or I'll update the description box with the PDF of the session. Is it clear for everybody? And what and how did they made out this particular DNA, rDNA, the new recombinant DNA? How do they made it out? They isolated an antibiotic resistant gene from the plasmid of a particular organism, Salmonella typhimurium. Very important MCQ. Okay, so the first recombinant DNA was prepared or constructed by Stanley Cohen and Herbert Boyer in the year 1972. They cut out a piece of DNA from the plasmid of an antibiotic resistant gene, from the plasmid carrying an antibiotic resistant gene in the bacterium Salmonella typhimurium. Clear? What is plasmid? Plasmid is an autonomously replicating extra chromosomal material. Clear? And what was a host over here? Plasmid is always used as a vector. Plasmid is always used as a vector, and it is transferred into the host organism, that is E. coli. You have to always remember two important things. Always in biotechnology, the host organism is E. coli bacteria, and the vector, what is vector? The carrier. Vector or carrier is the plasmid. What is plasmid? It is autonomously replicating DNA. Okay, is it clear for everybody? So recombinant DNA technology first was proposed by Stanley Cohen and Herbert Boyer. They cut out a piece of plasmid DNA from the antibiotic resistant structure of Salmonella typhimurium, and they link the plasmid vector with the transferred into the host organism, that is a E. coli bacterium. Okay. Yes. Next, we are moving on to the tools of biotechnology. I hope I have told each and every point that can come as MCQ from up to the portion topic over here. Next, we are moving on to the tools of biotechnology we are discussing right now. Okay. The tools of biotechnology, there are mainly three main tools of biotechnology. That is, the first one is the restriction enzymes. First one is the restriction enzymes. Okay, restriction enzymes, that is, they can cut out the DNA. They can cut out the DNA either from end, or they can cut out the DNA from a particular sequence. Okay, they can cut out the DNA from the ends, or they can cut out the DNA from the sequence. This particular restriction nuclease, restriction enzymes, can cut out the DNA into different small fragments. As per the requirement, this particular restriction enzymes can cut out the DNA into small fragments. Second one is the cloning vector. What is vector? Tell me, what is the cloning vector used in biotechnology? What is the cloning vector used in biotechnology? On the chat box. I have already mentioned you which is a commonly used cloning vector in biotechnology. Is it E. coli? Is E. coli the vector? No, you have understood the thing. E. coli is the host. Okay, E. coli is the host. What is the cloning vector? Vector means what? Vector is the carrier. Vector means what? Vector means it is a carrier. Vector means it is a carrier. So from where you are carrying the segment of DNA? Through the plasmid. Okay. Is it clear now? These are the tools of biotechnology. Restriction enzymes, cloning vectors, and the competent host. Okay. So cloning vector is actually the plasmid, and the competent host we are using over here is the E. coli bacterium. Clear? Is it clear? Carrier can or cannot be a gene from the host. It is not specified, or it's not specific that the plasmid have to be taken from the particular E. coli cell itself. Okay, it can be a plasmid from some other organism, and I told you that there are different methods of transferring of this particular gene of interest into the host cell. Can you hear me properly? Okay. For example, you can inject the plasmid into a host cell. You can fire a plasmid into a host cell. There are different methods. So just remember this particular thing. Vector acts as a carrier of the DNA segment. Host is the organism that receives the particular thing. Clear? Always the host in the biotechnological process is the E. coli, and the cloning vector is the plasmid. That's the only thing that you have to remember over here. Let's move on with the next one. The first tool of biotechnology is the restriction enzymes. Okay, the first thing is the restriction enzymes. Restriction enzymes are the enzymes which cut DNA at specific site into fragments. I already told you what is the function of restriction enzyme. They always cut the DNA to form fragments. Yes, vector carrier. Vector acts as a carrier. That is, it carries DNA segment from one place to another. And host is the organism that receives that particular cut DNA fragment or that segment. This restriction enzyme belongs to a particular class of enzymes called nucleases. Restriction enzymes can be mainly of two types. That is, first one, exonucleases. Exonucleases and endonucleases. Okay, exonucleases and endonucleases. For example, you can cut the DNA over here. Okay, you can cut the DNA over here. You can have a cut in the DNA over here, right? So this particular DNA cut can be made with the help of what? Restriction enzymes. Therefore, they are known as molecular scissors. Okay, restriction enzymes are referred to as the molecular scissors. Yes, they are of two types, that is the exonuclease enzymes and the endonuclease enzymes. Okay, so restriction enzyme is a particular tool used in biotechnology. They are of two, they belong to a large class of enzymes called the nuclease. They can be of two types, exonuclease and endonucleases. And this restriction enzymes, you can call it as the molecular scissors. Next, the cutting of the DNA fragments. Okay, I'll take a blank slide. Yes. The cutting of the DNA fragments can bring about two different things. Okay, I'm telling about the action of restriction enzymes. Sometimes the enzymes can cut the DNA into small fragments. Okay, the restriction enzymes can cut out the DNA into small fragments. Look, this is actually referred to as the blunt end. This is referred to as a blunt cut or blunt end. I'll tell you about the second type. Look out the second one. Okay, this is one type of. And the other one, look, here there is a side for the attachment. Okay, this can be placed over here very comfortably. So this particular thing you will call it as the sticky ends, because the two ends or the two fragments can be attached together with the help of hydrogen bonds. In the previous diagram, there is shown very clearly about the hydrogen bonds projecting out. Okay, when these two particular things can be fixed together, I'll show you what they will form. This is one particular sticky end produced. Okay, this is one particular sticky end produced, and this is another sticky end produced. Look, this is acting as a sticky ends. Okay, mainly for biotechnological process, we are using restriction endonucleases or restriction enzymes that produces a sticky ends instead of blunt end, because it is very difficult to rejoin or to connect between the fragments of DNA having a blunt end. Is it clear? Look, these are the hydrogen bonds. It's clearly mentioned these are the hydrogen bonds projecting out, and this is an example of a sticky end producing restriction enzymes. Here they have mentioned that these are enzymes which cut DNA at specific site. What is a specific site called as? The specific site, that is the palindromic sequence. Okay, the palindromic sequence. Palindromic sequence. What do you mean by a palindrome? For example, when you take the term Malayalam, this is the example mentioned in your NCERT also. Malayalam, when you take about, you can read it once. If you kept the site of reading like this, it is the same spelling. If you read it from back to the front side also, it is the same. That is referred to as a palindromic sequence. Another one, mom, you can tell, it can also be, if you kept the spellings of reading either from both sides, it would be the same. Okay. Yes, absolutely. That is an example of a palindromic sequence. Let me check how many slides are left about. Yes. Next, we are going to discuss about the conditions or restriction enzyme, which can also be called as molecular scissors. They can also be called as the Molecular scissors. First point is molecular scissors. They cut DNA at specific point. Very important. In between, they cannot go and cut. They cut the DNA only at specific points. Clear? Second one, the vector used over here is the plasmid. The host is the E. coli bacteria. The host used over here is the E. coli bacteria. Okay. Cleaving DNA makes small DNA fragments with sticky ends which are easy for us to rejoin them back with the help of DNA ligase. What do you mean by restriction enzymes actually cut the DNA, right? They are called molecular scissors. They can be reunited. They can be reunited back to form a recombinant DNA with the help of enzyme DNA ligase, which is actually, you can call it as a molecular glue. Okay, DNA ligase, you can call it as a molecular glues. These sticky ends allows the foreign DNA into the vector DNA and the double-stranded cut producing the blunt end. Okay, blunt end, I told you certain fragments like this which is very difficult to rejoin them back even though you use DNA ligase enzyme or any kinds of molecular scissors. Give me thumbs up on the chat box if you are clear up to the topic over here, and can we move forward with it? Yes. Look, the restriction enzymes cut the DNA at one particular site. Okay, instead of simply cutting the DNA like this, they will cut the DNA only at one particular site. Clear? So, moving on to the next one. I already mentioned to you about this particular thing, that is the restriction enzymes are of two types, exonuclease and the endonuclease. Restriction enzyme belong to a large class of enzyme which is called nucleases. They can be of two types, exonucleases and endonucleases. Exo means exit or the end portion. They remove nucleotides from the ends of DNA. Look, if this is a DNA template, okay, if this is a DNA template, the exonuclease will cut the DNA from the ends. Okay, look, they are producing the DNA from the ends. Whereas when it comes into the case of endonuclease, they cut the DNA at specific position, which you will call it as a palindromic sequence. Okay, definite palindromic sequence they will be forming, and they will cut the new DNA fragments at specific positions only. They can bind to specific recognition sequence of the DNA and can cut each of the two strands at specific points in the sugar phosphate backbones. Clear? All the things we are using, for example, a particular thing I'll write over here. For example, the palindromic sequence that identifies the endonuclease, one of the endonuclease is GAATTC. The palindromic sequence can be CTTAAG. Okay, so this particular endonuclease cuts the DNA fragments just like this. It cuts the DNA at a one particular fragment in the sugar phosphate backbone. Clear? So that it leads to the production of sticky ends. In between only they will cut instead of cutting the DNA at the end to produce the blunt end fragments. Next, one of the common example of a restriction enzyme is EcoR1. It's not EcoRI, it's EcoR1. Okay, you have to study what each of these terms reminds us, or how the naming of these terms is done. Clear? For example, the first letter of all the restriction enzyme, you have different restriction enzyme. For example, you have a restriction enzyme HIND2, SAL1, PUV2, then PSI1, Okay, so all these are different types of restriction enzyme, and now I'm going to explain you with the help of an example, that is ECO-R1. Okay, so the first letter of this particular restriction enzyme indicates the genus name. Okay, index indicates the genus name, that is the E stands for the genus name, that is it is derived from the genus. Okay, genus name, that is E stands here for the Escherichia coli. Okay, the second two terms stands together for the species. The second two letters indicates a species of the prokaryotic cell from which they are isolated. That is, E. coli bacteria is the host organism for this, or from the organism where this restriction enzyme is obtained. So E. Escherichia, co stands for coli. And what is R? R stands for the strain of bacteria. You might have learned about different strains of bacteria. It can be a wild strain, it can be a symbol strain, a gram positive strain, gram negative strain. So there are different strains of bacteria which you will study in detail in your higher classes. Okay, so R stands for the strain of the bacteria, and Roman numeral 1 stands for the particular order in which the enzyme can be found. There is an enzyme, there is an restriction enzyme named as ECO-R2. Okay, ECO-R2, because this is termed as 2 here because it is discovered secondly. This is discovered secondly. Okay, so the order in which the enzymes were isolated from the strain of bacteria. This is a previous year MCQ question. They have asked you ECO-R1, in which CO stands for coli, colus, and all these things. So it stands for coli. E stands for Escherichia E. coli bacteria. Clear? Is it clear for everybody? So first letter indicates the genus name, second letter indicates the species name, third letter indicates the strain, and the last Roman numeral indicates the strain from which the enzyme, the order in which the enzyme has been isolated. I think the last two slides we will discuss in the next class. Okay, in the next class we will discuss about the palindromic sequence and the DNA ligase action in detail. And now ready for a quiz, immediate quiz. Tell me, comment me the answers on the chat box. Okay, based on today's class I have prepared a quiz over here, so answer to the questions fast on the chat box. You would get 30 seconds to answer this question. Let's move on to question number 1 on your screens. Question number 1 on your screens. The core technique. Oh, the timer is not getting starts. Yes, you can see the timer over there. The core techniques of modern biotechnology are genetic engineering, chemical engineering, both A and B, none of these. Yes. 30 seconds to answer this question. Yes. Just think once again. We have discussed only about genetic engineering in today's. Okay. Clear. But we could say that both genetic engineering and chemical engineering can be the core techniques of modern biotechnology. Okay. In this particular chapter, in this particular 12th standard basic, we can study only genetic engineering in detail. We don't have to study about the chemical engineering. So please study, please note that the correct answer for the question is option number C. Both genetic engineering and chemical engineering can come as the core techniques. I haven't explained you about the chemical engineering. The modern biotechnology mainly consists of two main core techniques, that is genetic engineering and the chemical engineering. Okay, genetic engineering means you can alter the chemistry of the DNA. Chemical engineering means you have to maintain a sterile environment. It is a particular kind of biology by which you can maintain a sterile condition for the organism to replicate and to produce new further progenies. So as per this, a unit of this particular chapter, you have to study in further detail. And now we are discussing about the DNA recombinant technology and all, which comes under genetic engineering. But chemical engineering also, when you go to your higher classes and take biotechnology as a separate branch of your degree sessions. Is it clear now? Move on to the next question right now on your screens. Let's move on to question number 2. Yes. Oh my God. Yes. Comment me the answer for the question. I think there is no need of a particular timer. Yes, guys, comment me the answer. Calvin, just comment me the answer on the chat box very fastly. Plasmid used to construct the first recombinant DNA was isolated from the bacterial species. Plasmid used to construct the recombinant DNA, that is, the first rDNA was isolated from which bacterial species? That is, Stanley Cohen and Herbert Boyer isolated the DNA from which species of bacterium? The correct answer for the question, I hope there is a network issue and he left. The correct answer for the question is option number B, Salmonella typhimurium. We have discussed in the class in detail. Stanley Cohen and Herbert Boyer isolated the DNA from the Salmonella typhimurium. Okay. Let's move on with the next question. Absolutely. Okay. Option B is the correct answer for the question. Let's move on to question number 3. Question number 3 on your screens. The term molecular scissors refer to recombinant DNA, restriction enzymes, tag polymerase, palindromic nucleotide sequence. Please don't confuse it with the E. coli bacteria. Recombinant DNA technology, the thing the bacteria used was Salmonella typhimurium. Soon after the session, go to your NCERT, read out the NCERT. Just comment me the answer for the question. The term molecular scissors refer to. Yes, absolutely the correct answer for the question is answer number B, restriction enzymes. Clear? Restriction enzyme, because we have studied in detail about this particular thing, restriction enzymes. Clear? Moving on to the next question. Question number 4 on your screens. Question number 4 on your screens. Which one of the following is not a tool of genetic engineering? Which one of the following is not a tool of genetic engineering? Cloning vector, restriction enzymes, foreign DNA, GMOs. Very fast answer for the question. Very fast. Absolutely. The correct answer for the question is option number D, GMOs. What do you mean by GMOs? Genetically modified organism. Okay, genetically modified organism. Clear? GMOs is genetically modified organism. So the correct answer is option number D. So that's all about today's session. Hope you have got each and every point clearly. It took nearly 1 hour to explain a very small topic of the chapter biotechnology. So the PDF of the session. Okay, very good. You can, for other question also, you can do for the same. Okay. So thank you guys. See you in the next class with the lecture number 2 of this session. Bye-bye. Take care. Have a good day. Read out the NCERT right now. Take 10 minutes of your NCERT. First 2 pages of your NCERT of the chapter biotechnology you have to read right now. Okay, first 2 chapters, first 2 pages of the chapter biotechnology principles and process. Then only take the rest. Okay, then only leave the class, take the rest. Okay, so bye bye. See you in the next class with supremely amazing session 2 of this chapter.